Okay, good morning, everyone. It's time 11 a.m. Eastern. How are you? I'm Cherry Kubota. I'm a professor at the Ohio State University Horticulture Crop Science. This is a monthly webinar series or indoor forum mainly for indoor growers and technology providers for learning science and technology behind the indoor agriculture practices. This forum, Indoor Ag Science Café, is funded by USDA, and then the group working members include myself and other professors at the Michigan State University, Purdue University, and University of Arizona. So if you are new to this group, welcome. And if you are continuous participants, thank you so much being with us as usual. And so today we have a special speaker talking about not a leafy green, production, but something else to apply leafy green production technique. And then we have January Cafe. It's also a special program focusing on technologies. And that cafe, just to give you a heads up, January 18th, 11 a.m., Tuesday, 11 a.m., we will introduce 4 companies. All of them are providing new technologies. Fluence, which is a lighting technology, CO2 Grow, which is a CO2 enrichment technology, water harvesting, which is recirculation of water or harvesting water from the air. And then the Unfold Bio, which is the breeding company specialized for indoor. So it's going to be a great one. So please look forward to that. With that, I'd like to introduce quickly today's special speaker, Dr. Nobuyuki Matoba, or Nobi Matoba. So Dr. Matoba got PhD in Kyoto University, and then I met him first time in Arizona State University when he was working in the Biodesign Institute. And then now he's a professor at the University of Louisville, Kentucky, and working on plant-made pharmaceuticals, vaccine and all that, important for human health. Using biotechnology approach. So I like to stop sharing my screen. Or maybe I didn't. Oh, yeah. Okay, sorry. I got confused. I stopped my screen here, and then I will have Nobi in the podium.
Hello, everyone. Hi, Dr. Kubota. Can you hear me?
Yes.
Great. Okay, so let me share my screen. Okay. And then, all right, can you guys see my slide? Oh, I hope you can listen. So yep. Can someone speak up? Okay, great. Thank you. Thank you for introduction, Dr. Kubota, and it's my great pleasure to introduce to you guys, and I'm very excited and also a little bit nervous because I usually only speak to my graduate students, those students at because I'm at the School of Medicine. But the reason why I'm talking here today, I hope you find it very interesting, is because we develop drugs using a indoor plant facility. So I changed my slide slightly, where in the announcement, I think that the title was Plant-Made Pharmaceuticals under Controlled Environment, but I put bio in front of pharmaceuticals. I'll tell you the reason shortly. Today, I want to talk about, since for this audience, I think it's important to introduce pharmaceuticals. So I'll start with that. And then Plant-Made Bio-Pharmaceuticals and a history and current status to introduce this exciting field to everyone in this audience today. And hopefully I'm really hoping that some of you become very interested in this technology and share your expertise, because we do need a lot of research still to make this exciting field more economically feasible and commercially successful. And then if time allows, I want to introduce projects at the University of Louisville BioPharmaceutical Research Unit. All right, so I just want to briefly touch upon what we do at the University of Louisville, and we have 3 drug products under development using plants. And one of them is a Lactic Body. It's a unique cancer immunotherapeutic agent developed in my laboratory at R&D stage, research and development stage. And the second is an Episar, which is a drug candidate for ulcerative colitis, a type of inflammatory bowel disease. And then we've gone through R&D stage and then now at the preclinical stage. And we have initiated communication with US Food and Drug Administration. And the third one is the Q-Glyphosate, where we have gone, one of the side project, an HIV prevention project has already gone to the phase 1 clinical trial stage, and we have just completed the phase 1 clinical trial in summer this year. And then we also have another COVID nasal spray prophylaxis. It's an alternative to vaccine to prevent infection against SARS-CoV-2 virus, the virus COVID-19. We have just initiated a new phase 1 clinical trial, and we actually had second cohort of participants coming to our clinical trials, and we are very excited about this project as well. But hopefully we have time to discuss this project, but let me start with the basics of biopharmaceuticals. So I'm not sure if you are familiar with biopharmaceuticals, but I just listed some of the COVID-19 drugs currently under authorized or approved. Can you guys tell which ones are classified as biopharmaceuticals? I'll just give you a second or two to think about it. If you have no idea, that's fine. I'm going to explain in the following slides. So dexamethasone is prevent excess inflammation, and remdesivir, second one, use my pointer. Remdesivir is an antiviral drug. Monoclonal antibodies also authorized, and they are from Eli Lilly and Regeneron against mild and severe, mild to moderate COVID cases. And as you know, vaccines are based on mRNAs, and then also adenoviral vectors. These are already authorized, and some of you have probably received one of these vaccines already. So I'll tell you the answers later. So what are biopharmaceuticals? They are pharmaceutical products that are produced in biological processes using biotechnological methods. They include vaccines, and then nucleic acid and gene therapies, engineered tissue and cell, and then recombinant proteins such as monoclonal antibodies. These are called biopharmaceuticals, produced using biological processes. And they are made of very complex macro and large molecules that have specialized biological functions. Right? Usually these molecules are derived from our own biological molecules, including the antibodies, right? So these are specialized to have special biological functions, and therefore they are very usually specific and therefore safer, and then they're also very potent. But the caveat or the downside of biopharmaceuticals are that they cannot be chemically synthesized. They are too complex to be synthesized in the laboratory. So just put it into context more. So take these two COVID drugs examples. So remdesivir I just mentioned, and then bamlanivimab, that's a monoclonal antibody from Eli Lilly. The weight of remdesivir is 0.6 kilodalton, whereas this monoclonal antibody is 144 kilodalton. The chemical formula, you can see the remdesivir is relatively easy to remember, perhaps, but it's impossible to even think about how big it is, right, for the monoclonal antibody. So if this is the remdesivir, the monoclonal antibody is something like this. So it's very difficult to produce, and then chemical synthesis is impossible. Therefore, we have to rely on biological systems. And these are the conventional biopharmaceuticals manufacturing platforms, including E. coli bacteria that are used for insulin and growth hormones and interferon productions. And then yeast cells are used for some of the vaccines. And then insect cell, moth cell, are also used for vaccine production and a type of influenza vaccine, not the one that is produced with chicken eggs, but there are some protein-based vaccines produced in moth cells. And then lastly, this is the mainstream in the industry, is the use of the mammalian cells, more specifically Chinese hamster ovary cells or other cells that are used to produce monoclonal antibodies, and then also erythropoietin and other protein-based drugs. These are all cell culture systems you can note, as you may notice, and therefore rely on the use of tank, something like shown like this. So, and then E. coli is a prokaryotic cells, and therefore are limited to produce relatively simple proteins and peptides. But yeast and insect and mammalian cells are eukaryotic cells, and therefore are equipped with machineries to make more complex and large molecule proteins, protein-based pharmaceuticals. So in 1982, over roughly about 40 years ago, the first FDA-approved biopharmaceutical reached the market. That was a recombinant human insulin. Before that, the insulin was produced from the pigs, and purified from pig tissues or the blood. How it's produced in bacteria using recombinant DNA technology. So since then, the biopharmaceuticals has been rapidly expanded in the pharmaceutical field. And in 2019, 2 years ago, within these top 10 best-selling drugs worldwide, seven of those 10 were biopharmaceuticals, selling $67 billion of $92 billion total sales in the world. And these seven biopharmaceuticals are including Humira and Keytruda. Humira is an anti-inflammatory antibody in arthritis treatment. Keytruda is one of the main immunotherapeutic agent for the cancer. It's a checkpoint inhibitor, otherwise called. And Opdivo is another checkpoint inhibitor in oncology. So you can see how biopharmaceuticals are currently mainstay and then main focus of pharmaceutical industry. So back to this initial question to you, probably you can tell now. So among these 5 COVID vaccine, COVID drugs, and these are the biopharmaceuticals. Right, with that, I'm moving on to plant-made biopharmaceuticals. I hope now at this time you can tell the definition of biopharmaceuticals. Again, those are the complex macromolecules produced in biological processes, right? And then currently bacteria, yeast, insect, and mammalian cells are used, but we propose to use plants. And why is that? Intuitively, I think you all can understand using plants can be very economical, right? And the first and foremost to build a mammalian cell culture facility for pharmaceutical production, like the one on the image on the right. And this costs like hundreds of millions of dollars to construct such a facility. But it's much cheaper to construct greenhouse facility and indoor growth facility. So therefore, the significantly less and small capital investment is required when we produce biopharmaceuticals in plants. And then what's more, the raw materials are much more inexpensive in plants compared to mammalian cell culture, which require very expensive materials and nutrients and other things. And the scalability is limited in the case of steel tank. This pretty much sets the batch size and production capacity. However, if we use greenhouse or the plant growth facility, the scalability is much more flexible. And last but not least, did I hear something? Well, yeah, I'm gonna move on. Yeah, but if you have any question, please speak up. You can ask me a question anytime if anything is unclear or you have question, please. So so the last but not least, the mammalian cell system has the risk of contaminating human pathogens and viruses and oncogenes, whereas in plants, there's no known human pathogen that can grow in plant cells. So there's a safety with plant system. So there are two major methods to produce biopharmaceuticals in plants. And the first and the more conventional method is the transgenic plants or genetically modified plants. And the second, more mainstream right now in the industry is the use of transient gene expression system. So let me first explain this transgenic plant system. So the transgenic plants or genetically modified plants were the first platform that was tested and used in biopharmaceutical production field as well, where the Agrobacterium tumefaciens is primarily used to transform plant cells, and the bacteria has a unique capacity to move and transform plant cell. The researchers have modified the capacity of the Agrobacterium to transform plant cells to introduce the gene of interest, gene that can that encodes for the protein in this case, such as monoclonal antibody. And same method has been used to generate herbicide-resistant plants or crops or insect-resistant crops, right? So instead of making insect-resistant crops that would express some toxic molecules for the insects, so we would introduce gene that would encode for for example, vaccine or monoclonal antibody, and then use infect plant tissue or cells, and then the plants can now be integrated into whole transgenic plants. Now the gene of interest is integrated into the chromosome, the genome of the plant cell, and thereby producing your protein of interest. So when tissue culture or transformation methods are available, pretty much any plant can be transformed and then turn them into a factory to produce protein of interest. Now, using this technology, more than 30 years ago, am I correct? Yeah, 33 years ago, the first example of producing antibodies in transgenic plants was reported. And then 3 years later, my former supervisor, when I was a postdoc at Arizona State University, Dr. Charles Armsten's group has, for the first time, shown that vaccine can be expressed in plants. And then his group has also subsequently shown that the vaccine antigen, or in this case, bacterial produced in potato plants, can be used as an oral vaccine. And then the volunteers has consumed this transgenic potato, and then showing that they can mount the vaccine can induce antibodies in the volunteer's blood that can neutralize the bacterial toxin. This research has been published in Nature Medicine in 1998, and is showing the proof of concept for edible vaccine idea. And by the way, I was very fascinated by this idea as a graduate student back then in Japan, and that's the reason why I wanted to move to this field and then do research under Dr. Charles Anzen. Anyway, so in the same year, 1998, there was another paper published where they have also performed a first-in-human study using plant-made antibody against the bacteria that causes tooth decay. And these two papers have shown proof of concept again that the plants can be used to produce vaccines or biopharmaceuticals. And then there are two products now have been marketed, approved for marketing. And one of them is an FDA approved drug. It's used for enzyme replacement therapy for a rare disease called Gaucher disease. So the Gaucher disease patients lack the enzyme in their body, but this company called Proteinbased Biotherapeutics, which is in Israel, developed a commercial enzyme produced in certain plant cell suspension culture system shown in this image. Can you tell what kind of cells are they? And based on the color, these are transient carrot cells. So they use this unique bioreactor system. Note that they use instead of steel tank, therefore making it much more cheaper than the conventional systems. In this case, however, of course, the enzyme needs to be purified for injection using as edible vaccine or anything. So after using this to produce an enzyme, they would purify completely, and then the purified enzyme is used as a treatment. And another product based on transgenic plants has reached the market in Japan. It's an interberry. This was approved back in 2013. It's for dogs, not humans, but still needs to be approved by a regulatory agency, and which they did. That was developed by National Institute of Advanced Industrial in collaboration with Hoxton company. They produce canine interferon alpha, which is an antimicrobial protein expressed in genetically modified strawberry. So they use strawberry in contained facility like shown in this image. But in this case, it's closer to edible vaccine idea where this antimicrobial protein is not purified, but rather the transgenic fruits would be freeze-dried and then powderized. And this powder is now applied to the gum tissue of the dog to treat this gingivitis. Now, the second method, which again, as I mentioned, this is the mainstream in the field right now, is a transient gene expression, in which the plants are not genetically modified. In fact, the regular plants are grown, but only when after they've grown to certain size, then the transiently introduce the genes into the leaf tissue and then expressed in plant tissues, and then the biopharmaceuticals are purified afterwards. So for that, the system utilizes plant viruses. Because if you think about it, viruses are adept at making a lot of proteins as part of their the components, such as tobacco mosaic virus, virus X, and Gemini viruses. they have very simple structure with relatively simple genome. They have the replication units, and then there are protein that is a shell that encodes and encapsulates their genome. So the idea is to exploit such viruses to put the gene of interest, for example, some interferon or bacterial toxin proteins as vaccine candidate as part of their genome. Then you would use that modified virus to infect plant called Nicotiana. It's a species of tobacco, Nicotiana family, and genus Nicotiana, and then it's an indigenous to Australia, by the way. Plant is used as a host, and then as the virus replicates, then your protein of interest would be also produced. So this is an example when green fluorescent protein is expressed using a modified tobacco mosaic virus. As the virus spreads through the plant systemically, you can see the green fluorescence is fluorescent protein accumulated in leaf tissue. But this first generation modified plant virus was suffered from the limitation, which was the size of the protein that can be produced as part of the virus was limited, because the virus needs to be still functional and then need to replicate and systemically moving to infect the whole plant. But that limitation has been solved by using a deconstructed virus vector system, where instead of using the whole virome, so we would utilize just the replication unit or replicon of the plant virus. And then that region of the replication unit would be transferred into a DNA vector of Agrobacterium tumefaciens. Remember, Agrobacterium is used to make transgenic plants or genetically modified plants. So this bacteria has the ability to move this vector DNA into leaf tissue. Now, instead of making transgenic plants, you vacuum infiltration called Aggro infiltration, and then thereby transferring this replication unit to the entire leaf tissue of the plant. So this is, well, I was hoping to show the vacuum infiltration video, but looks like
Dr. Matoba, I think you need to switch the laser to regular cursor.
I see. So, there you go. Thank you, Dr. Kubota. So this is how vacuum infiltration works. So you can see this is an enlarged image from this chamber here, where plants are put upside down. And this liquid part is the bacterial suspension, the water containing bacteria and other buffer. You can see as the vacuum is pulled and then released, the bacterial solution is incorporated into leaf tissue through stomata. And each bacterial cell would contain this that contains replication unit from the virus. And therefore, the entire leaf tissue now can become factory to produce your protein of interest. Okay, that went well. Back here. So yeah, this process is called Aggro infiltration. And now this is compatible with industrial production, which is shown here. So this is an overview of biopharmaceutical production in Nicotiana bentamiana transient expression system. So where the plants are grown indoor up to 4 weeks, and then plants are moved, turned upside down, and then dipped into this agrobacterium solution chamber, and pull vacuum and release, and then plants are stuck into the incubator space, and then grown and incubated for 1 to 2 weeks. And then after that, the leaf tissues are harvested and then leaf proteins are extracted. Then the extracted proteins that contain all sorts of other contaminants and plant-derived proteins, in addition to your biopharmaceutical protein of interest, therefore it needs to go through extensive purification process using chromatography. And then finally, you analyze the biopharmaceutical after purification, and then that becomes product. Now, using this system, in 2010, a Canadian company called Medicago had shown that they can produce avian influenza vaccine, and then that received emergency use authorization from FDA. 4 years later, Kentucky Bioprocessing, another company, in collaboration with BioPharmaceuticals, which is in San Diego, they co-developed an experimental Ebola virus antibodies, and then that was used in a compassionate basis in West Africa, showing that those who received the plant-made antibodies dramatically recovered from Ebola virus infection. That was featured in many news media, and it was a very big news for our field. And they've also completed phase 2 clinical trial back in 2016. And then last year, again, the Medicago has completed the last stage, the phase 3 clinical trial for seasonal influenza, and showing promising results. And finally, COVID vaccines also produced in Nicotiana bentamina plants by Medicago. Medicago finished phase 3 clinical trial, and then they're awaiting regulatory approval from Canada, and they're also going to be, they seek authorization in Japan next spring. And this is a news I found this morning. And the Medicago announced that their vaccine is effective against Delta variant by 75% efficacy. And then, so they are hoping to receive authorization any minute or any day now. So about this vaccine against COVID, I just want to also mention this. So as you know, mRNA vaccine has been already used widely, but there is not any protein-based vaccine available in the US yet. But these are the companies that are the front runners producing protein-based COVID vaccines. Protein-based vaccines can be advantageous over mRNAs in certain situations because they can be stored and then transported at much mild conditions, refrigerated conditions. And some people may prefer more conventional protein-based vaccines than mRNA or adeno vector-based vaccines. You can see Medicago is one of the top front runners amongst those other companies that are producing protein-based COVID vaccine. But I just want to ask you, what else do you notice in this table? So these are all protein-based. Now, if you look at the cell manufacturing system, so Medicago, of course, use tobacco plants, Nicotiana bentamina, but others use, as I explained, they use conventional systems like yeast, mammalian cells, moth, mammalian, right? So this illustrates that the plant-based Nicotiana bentamina transient expression system can now be as competitive as conventional systems. They can produce vaccines as quick as the other conventional systems, and then can become marketed like other vaccines produced. And then among these companies, Novavax vaccine seems to be the first one to be approved or authorized. But as I said in the news, as you saw, the Medicago vaccine will be also authorized very soon. So As far as I know, there are only 3 commercial scale facilities in North America that use Nicotinamide bentamine transient expression system, including Medicago. Their headquarters in Quebec City, but they also have facility in North Carolina, and they are making also a big facility in Quebec City. And they use natural light greenhouse for vector inoculation, and then the plants are then moved into indoor for the additional incubation after Aggro infiltration. Kentucky Bioprocessing, on the other hand, use all indoor facility for the plant growth and incubation before and after Aggro infiltration. And there's another company called in Texas. They have a very big, humongous indoor facility. And you can see that they use different lighting systems, which may be of interest to some of you. So the system has not been standardized yet, and then there's much room to optimize in that context. So I just want to talk about this current good manufacturing practices. So what are these? So CGMPs are regulations that govern the manufacture of human and veterinary drugs, biologics, medical devices to ensure the quality of the finished product, right? So this is a Code of Federal Regulation numbers. So why this is important? Because drug manufacturers are required to fully compliant with the CGMP if they are making drugs for humans or the animals, right? And then in fact, the FDA would come and inspect your facility if you're making drugs or drug candidates, and then they can always suspend your approval or authorization if they find anything wrong. So why this is important is that the quality, of course, of drug product is very important, but the quality cannot be only tested or inspected into the final product. In fact, the quality needs to be built into the whole process of manufacturing, including materials and methods and facilities and equipment, and all these processes and controls, right? So to put this into the context of plant-made pharmaceuticals, let's just look into the whole process of the plant-made pharmaceutical manufacturing. So here showing the cartoon image showing the schematic flow of the transient expression system, starting from so Aggro infiltration is here, and then you have pre-infiltration plant growth, post-infiltration plant growth, followed by extraction, purification, characterization, formulation, as I explained in a few slides back. So this process can be divided into 2 upstream and downstream processes. So from FDA standpoint, currently, the CGMP only applies after the extraction, and this process number 4, 5, 6, 7, after 4. However, still FDA requires the upstream process and the extraction to be fully documented and then controlled. And then each step requires optimization, tight control, and monitoring, and or method qualification validation to meet the regulatory requirements. Right. So you can't just grow plants and without any control or documentation. You have to follow procedures, and you have to keep records on how each batch is produced. So the importance of the plant growth conditions from the regulatory standpoint. So this is based on our own experience of going through phase 1 clinical trials. Whenever we communicate with FDA, they require the following information to assure product quality during the upstream production process. So one of them is the risk assessment for potential adventitious agents that may be introduced during the manufacturing process, such as pathogens contaminated in fertilizer or growth media. They want to know how this is controlled and how this is monitored. and then also strategy to prevent exposure to environmental pests, and then procedures and tests to monitor and ensure plant health. Why plant health is important? Because if plants are sick, not healthy, the proteins or biopharmaceutical produced in the plant would also be compromised. It's again, biopharmaceuticals are complex macromolecules consisting of amino acids and sugars and lipids sometimes, and then they can be oxidized or degraded, or some other chemical modification may be introduced. So therefore, keeping plant healthy throughout the process is very important. So to that end, indoor facility is crucial, because indoor facility can provide physical containment to assure security of manufacturing environment first and foremost, and also to provide controlled environment to prevent gene flow. Recombinant DNA materials are not allowed to go outside the facility, right? And the indoor facility can help prevent contamination. Again, the FDA cares about pathogen and insect contaminations in the process, and the use of the indoor facility would minimize the use of pesticides. If any mold or any pathogens are found in a facility, you have to completely decontaminate, and that needs to be also documented and then recorded if that happens. And then for the stable and uniform production, indoor facility would ensure consistent product quality from batch to batch. So you have to continuously produce the same vaccine or antibodies or anything. So you have to keep the quality between batches consistent. Otherwise, some lot or some batch of the drugs may be less efficacious, or sometimes maybe less safe. That is not acceptable. And finally, this is not about the FDA's quality regulation, but optimization of plant growth environment is important to maximize productivity, right? So the more pharmaceutical proteins you can make from a unit area, the more profitable it is from the commercial standpoint. So this last point, this is also under investigated area, if you will. So I just want to touch base on the efforts currently by my collaborator, Dr. Ryo Matsuda at Tokyo University. He is looking specifically into optimizing indoor plant growth conditions to maximize productivity of biopharmaceuticals. So focusing on this upstream, he has been doing very productive research on pre-infiltration conditions, including light intensity, temperature, and gas, plant density and fertilization, also vacuum infiltration conditions, and post-infiltration conditions. And then also wrote a review article a few years ago. And just to briefly introduce what he's found in his research, is that optimal plant growth conditions are different between pre and post infiltration phases. This is very interesting, but what he found is that this is just data showing the amount of hemagglutinin, which is an influenza vaccine, right? So the y-axis is the amount of influenza vaccine produced in leaf tissue, and the x-axis you can see different temperatures. In the pre-infiltration growth conditions, it appears that the higher the temperature within 20 to 30 degrees, the more vaccine is produced in leaf tissue. On the other hand, post-infiltration, it's now shaping where the high temperature is counterproductive, and in fact, somewhere around 20 degrees seems to be optimal for the accumulation of the vaccine protein in leaf tissue. So what he found is that under pre-infiltration conditions, the focus should be on biomass accumulation. The more biomass, the more productivity, for which the relatively high temperature seems to be optimal. But once Aggro infiltrated, and now you don't want to put the plants in high temperature anymore, and then the main focus should be on mitigating the stress. The plants are stressed as producing massive amount of vaccine protein in their leaf tissue, right? For which apparently lower temperature is better. So perhaps in the near future, Dr. Master can come to this cafe and then introduce and then talk more about his research. All right. So Dr. Kubota, I don't know how much time I have. I just very briefly touch on what we are doing. So we
please go ahead. Go ahead.
Yeah. Okay. All right. So the current project. So again, so we have 3 projects. I just want to briefly introduce this Lactic Body project, which is an early stage, and then the Q-Glyphosate project, which is currently in phase 1 clinical trial. So the Lactic Body is a protein derived from actinomycete bacteria. It's a derivative of actin-inhibin protein, which is the crystal structure of the protein shown here. It's a small lectin or sugar binding protein derived from this bacteria. And interestingly, this molecule, this protein, binds to unique carbohydrate structure found on viruses and cancer cells. In other words, many viruses and then cancer cells in our body have very unique sugar structure on the surface. And this has been found in the recent years and then has been the target of potential therapy. And we utilize this protein to target cancer. That's how our project was initiated. But what we soon found was that this protein is very poorly soluble in aqueous solution and therefore can be expressed and produced very poorly in plants. So what we did was to take advantage of the speed of the transient expression system, where we did an in silico design of the designing mutants to reduce hydrophobicity, increasing solubility in water. And then we made a bunch of different mutants and then expressed in leaf tissue using, in this case, instead of doing a vacuum, we did a syringe infiltrated into each leaf for each mutant, and then screened SDS page analysis. This is an analysis to quickly monitor what kind of proteins are in leaf tissue. Each lane is different leaf extract protein, if you will, and then actin-inhibin. So here the showing is the molecular size. The actin-inhibin be expressed in this area. So each band represents different proteins in this case. This is the control. This is just a vector long. Actin-inhibin is very poorly expressed, but you can see one of the mutants is very thick, meaning that this protein, this mutant is highly expressed in leaf tissue. And we named this variant Avian, actin-inhibin variant, expressed in Nicotiana. So we made this variant. Now we further modified to create a chimeric molecule between human antibody and Avian, creating a Lactic Body, lectin antibody fusion protein. This was highly expressed in Nicotiana bentamiana and then exhibited very potent activity based on this, putting 2 molecules together in one, two into one molecule, in other words. And then we've shown this molecule has very potent activity against hepatitis C virus in humanized model. And furthermore, we've shown recently that this is a PET-CT analysis, positron emission tomography or computed tomography analysis of mice that has melanoma tumors right here, there is a melanoma in this mouse model here. And when we used Lactic Body that is labeled with kappa 64, we could clearly visualize the tumor, indicating that this Lactic Body is very specific to tumor. And this is, by the way, the liver, which is a metabolizing organ that always shows up in the PET analysis. But you can see, other than the liver, we can clearly visualize the tumor, again suggesting the high selectivity to tumor. And then we used human lung cancer models, mouse models, showing that the tumor volume after days of the tumor challenge, our drug, Avian FC, is a Lactic Body, can retard or prevent the tumor growth, as well as one of the FDA-approved immunotherapy agent. And another lung cancer model where FDA-approved drug has no effect, but our Lactic Body has shown complete protection against this tumor. And these results suggest that the Lactic Body could be a new anti-cancer agent. And we are doing more research to further develop this agent. And then the second one I want to talk about is the COVID prevention using Q-glyphosate. Q-glyphosate is another sugar binding protein, but this one is derived from lead algae called Glyphosate. And it binds to another sugar molecules on different viruses, including HIV, influenza, and SARS-CoV-2, the virus that causes COVID-19. And then by binding to this protein can prevent entry into human cells. And Q-glyphosate is a modified glyphosate, which is resistant to oxidation. And then now this molecule is efficiently produced in Nicotiana bentamiana using tobacco mosaic virus, and very potent anti-SARS-CoV-2 activity. This value, the lower, the more potent against the virus. But this molecule has different antiviral activity against different coronaviruses and then HIV. So it's a broader spectrum antiviral protein. And then we haven't seen any toxicity in animal models. So this phase 1 clinical trial program the purpose is to offer an alternative preventative measure against SARS-CoV-2 infection for high-risk individuals, for those who cannot benefit from vaccines. Vaccine doesn't induce antibodies against the virus, or some other high-risk conditions, or those who don't want to take vaccines. This can hopefully provide some temporal but effective prevention against infection. So we want to develop a nasal spray like this, put in a bottle like this. And then this phase 1B, phase 1A clinical trials are funded by DOD. And primary endpoints, phase 1 trial is safety and tolerability and pharmacokinetics. And this is the project leadership team. The overall project leader is my colleague, Kenneth Palmer, and I'm doing analytical quality oversight. And we collaborate with clinicians on campus and a regulatory consultant and a formulation scientist. And the Q-glyphosate was produced by Kentucky Bioprocessing using the tobacco mosaic virus, modified virus system. And the one of the production campaign, using over 200 kilograms of leaf materials, nearly 6,000 plants, and we obtained over 99% pure Q-glyphosate, final yield of nearly 400 milligrams per kilogram of leaf materials. So we got about 80 grams total. And then we submitted a document to FDA compiling all the preclinical data, toxicity data, efficacy data, and then we received FDA study may proceed letter 2 months ago, and the study has initiated at our hospital. And so far we've recruited 14 healthy volunteers, and we are planning to recruit 18. So first cohort, there were 6 volunteers. And this week, actually yesterday, I went to the clinic and the additional 8 volunteers came. We haven't seen any no report for any product-related adverse effects, which is great, very good news. And then based on this phase 1A, which is just a single dose, and we are planning to do multi-dose study soon after the completion of this phase 1A. Yeah, so those are just a brief overview of what we are doing. So early stage and late stage. So the reality, however, in the academic drug development programs are that after phase 1, we are not able to continue development. We have to rely on industry partner because it costs much more money and it needs more number of patients or volunteers. And then that becomes beyond the scope of academic institutions, and both in terms of financial and resource perspectives. So we are looking to find commercial partner to develop after phase 1. But so I just want to summarize the seminar today. So number 1, the biopharmaceuticals are complex macromolecules produced in biological systems, processes, and then they are rapidly growing in pharmaceutical industry. And I could convince you all that plants can offer unique and scalable biopharmaceutical production platforms. And in addition to scalability, the transgenic plants may provide some economical advantage, particularly when you don't have to purify the drug ingredient. And on the other hand, transient systems can allow rapid research and development like Medicago has done, like we did with Lactic Body. You can use that system to modify any protein of interest and do protein engineering and develop the drug candidate very rapidly. So these can be added to the scalability benefit of plants. And then we hope to see a new COVID vaccine, protein-based vaccine made in Bentamiana plants, and then that will be authorized very soon. And then lastly, I want to emphasize again that the controlled indoor plant cultivation is very important for the upstream production of biopharmaceuticals. I just want to acknowledge and say, thank my collaborators and Dr. Hamorowski and Palmer and Dryden and Guo. And Ryo Matsuda, again, he's my good friend and long-term collaborator. And Daniel Tousse for regulatory consultation, and Kentucky Bioprocessing for the production, and Icon Genetic for the transient expression system vector, providing vectors for us. And the funding was, support was from Helmsley Foundation and NIH and DOD. And then this picture shows my current group members. Matt has recently defended PhD, and he's looking for a job in pharmaceutical industry. And there are 2 technicians and a postdoc, and Wendy and Katarina and Mickey are students. Okay, with that, thank you very much for your attention, and I'm happy to take any questions.